My name is MJ Hopkins. I am currently a senior at St. John's University. I'm studying biology and psychology as a minor, and I am on the pre-med track. This summer, I was able to go to the University of North Carolina, Chapel Hill via HuBMAP to study the characterizations of basal cell phenotypes in the human lung, working in the Haygood lab. So first, I kind of want to start off and talk a little bit about what basal cells are and why they're important, as they're the main focus of our project. Then I want to go into a little bit about the goals of our experiment and talk a little bit more about how we achieve those goals and the methods we use and the antibodies we use. Lastly, I'll talk about the results and our conclusions that we got from the summer. So overall, the first thing we need to know is what basal cells are. So basal cells are progenitor cells found in the airways of the lungs. They differentiate into different cells based on the location and interactions within the lung. The initial analysis of the HuBMAP data done by Elizabeth Duong at the University of San Diego found that basal cell subsets that are usually found in the proximal airways of our lungs are being found in the distal regions unexpectedly. Here we can see that basal cells are not only progenitor cells, but they're also classified as epithelial cells. This is just an example of what the basal cells can differentiate as in the different cells that they can differentiate as in the lung. And we can see below in red the initial question posed by Dr. Duong. So the goals of our experiment, my initial goal going in was to see what the correct concentration of antibodies used could be able to determine the characterization of the basal cell in the airways that we were using. The initial goal for the parent HuBMAP project is to optimize cell recovery and that are being performed in single cell and single nuclei techniques while retaining morphology, relationship to physiology, and translation back to organ. We are also trying to discover which antibodies will be most useful in identifying basal cells subsets throughout the different areas of the lung, while gaining insight to how location of basal cell subsets correlate with airway histology, and to find the correct concentration of specific antibodies for subsets of airway basal cells. Ooh, sorry about that. So the methods that we used for this project was immunofluorescent staining. So the first part of that is to obtain the samples that were donated to us by the University of North Carolina at Chapel Hill Hospital. We then fixed the tissue in FFPE, which preserved the samples. After fixing the tissue and applying it onto a slide, we were able to apply the primary antibody, which we allowed to set overnight, and then applied the secondary antibody the next morning or the next day, which we allowed to set for an hour. After those were both applied, we applied a mounting media using vector cover slip with DAPI and applied the cover slip for imaging. The reason why the concentrations are so important is that we have to be able to find the correct wavelengths that we are working with. So we worked with size 7 and size 5. Both of those were used to determine which antibodies we were working with. So size 7, we used the wavelength of 750, while size 5, we used the wavelength of 647. Size 7 has the wavelength range of approximately like 650 to approximately 850, while size 5 has the wavelength of approximately 550 to around 650. The antibodies that we used were Ki-67, which is a nuclear antibody, Serpin B13, which is both cytoplasmic and nuclear, and keratin 5. These 3 antibodies were determined by Elizabeth Duong's lab and was given to the Haygood lab, which I worked in. So the results that we got from the Ki-67, as you can see, the first photo is originally taken from the lung map. You can clearly see the Ki-67 noted as a nuclear agent. And the second photo is what we obtained in our lab. And you can see that there is some overlap between the DAPI and the Ki-67. The results for keratin 5 or for K5 was really good. We were able to find the cytoplasmic nature of it. As you can see in the second photo, the green K5 is surrounding the DAPI, which is a great thing. And in the first photo, I just removed the DAPI so that we can clearly see the K5 cells. Lastly, for SERPIN-B13, we were able to see some overlap between the DAPI and the SERPIN-B13. As I stated before, SERPIN-B13 is both cytoplasmic and nuclear. So we can see how the SERPIN-B13 is able to overlap the DAPI as well as surround it in both photos. So for our conclusions, we determined that tissue samples that were inflated with optimal cutting temperature, which is OTC, before sectioning allowed for clearer airway results and better visualization of the cells within the airways. The concentrations that we were able to obtain for SERPIN-B13 was 1 to 50 for size 7 and 1 to 100 for the secondary antibody. For Ki-67, it was around 1 to 100 for the primary antibody, also used with size 7, and 1 to 150 for the secondary. And for K5, it was 1 to 100 for the primary, size 5, and 1 to 400 for the secondary. Moving forward, we will continue to use HuBMAP, staining the HuBMAP sample blocks using these antibodies and concentration while continuing to optimize the protocols that I learned throughout the summer. We also will begin to test different antibodies, additional antibodies, to determine basal cell subset interactions with other subtypes and to continue to find those additional antibodies concentrations. I would like to thank the HuBMAP Consortium, the NIH. Thank you guys for this amazing opportunity for me to go down to University of North Carolina. I learned a lot and I am forever grateful for that opportunity. I also like to thank my PI, Dr. Jim Haygood, and my bench mentor, Dr. Lorraine Riviere. I had a great time and I learned so much from both of you. I would like to thank Elizabeth Wong for allowing me to be a part of her project, and Dr. Gloria Pryor-Huber for everything that she has done in addition to this project. Thank you.